Hello. Welcome to another lesson. Today I'm going to cover protein synthesis. I'm going to cover those three stages which are required for the formation of proteins. And I also give sort of like examples to show you guys how you can go from DNA to RNA and then later on form amino acids. And then also how you can go from RNA back to DNA, and also how you can go from tRNA to DNA. So I'll do some practical examples just to show you guys how to go about those problems. So first thing first, remember so far we've discussed DNA and RNA. So there's a process called central dogma where we go from DNA to RNA, and then from there we also go to proteins. They call it a central dogma. You go from DNA, then go to RNA, then go to proteins. And remember, the aim of protein synthesis is to form proteins. So we need to go from DNA to form proteins. So also from grade 10, they told us that the building blocks of proteins are amino acids. There was always that question, one word question, that they kept on asking grade 10s about the building blocks of proteins, which are amino acids. And there were other questions like building blocks of fats or carbohydrates, which we won't focus on. Here, our aim is only proteins. So whenever you've got two or more amino acids combining, this process is called dehydration synthesis. So during this process, we've got two or more amino acids combining. If you've got two amino acids combining, two means two. So the process is called dipeptide. We form a dipeptide. If we have two, three or more amino acids combining, remember from also grade 10, poly means many. So if you've got three or more amino acids combining, then we form a polypeptide. And crucial, because I've seen a question or two where they're asking about the inorganic substance which is formed during dehydration synthesis. So whenever we're combining two amino acids or more, water is released. So always become aware of that product, which will be water, in case they would ask you a one-word question or any other question in a long question. So now the three stages which I want us to focus on: transcription, where we form messenger RNA. Then we've got the movement of the same messenger RNA out of the nucleus through the nuclear pore. And then we've got the last one, which will be where we're going to form proteins using tRNA. So here I'll just show you a quick diagram of how transcription will result in the formation of messenger RNA. So let's say this is my DNA sequence, because I can see that it's a DNA because we've got thymine. So I see T. Whenever you remember, DNA has got thymine. There's a thymine here, which means this sequence is DNA. Here, there's no thymine. We only have uracil. Remember, uracil is on RNA. So by virtue of that, I know that this is my DNA sequence. This one becomes my RNA. And remember also, guanine always complement with C, cytosine, and we know adenine always complement with thymine, but there's no thymine in RNA. So instead of thymine, it becomes uracil, which is U. So always become aware of that. If I'm having a DNA sequence and it's going to messenger RNA, there is no T in RNA. Because whenever you make that mistake, I normally just mark the letters 0 or just cancel because it does not exist. So again, we know there's thymine in DNA. So if I'm having RNA as A, we know in DNA I can have T. So don't get confused now. And then if they give you mRNA, then they're asking you to go back to DNA. You can't write U in DNA. There's no U in DNA. So it's only T. So the difference here, G and C will remain the same. The difference is that in DNA, we've got T. In RNA, we've got uracil. Remember, once you form the messenger RNA, it then goes out by the nuclear pore. That's the whole process of transcription. So this process of transcription, it requires a transcriptase enzyme, which is called RNA polymerase. This RNA polymerase is a very, very, very strong enzyme. Some of you will be studying more about HIV maybe next year. There's what we call reverse transcriptase, where the enzyme works in an opposite direction. So here also for transcription, we need RNA polymerase. So also become aware, guys, whenever you've got triplet of bases, we call them, if they're in mRNA, the triplet bases which are in mRNA, we call them codons. Then the triplet bases which are in tRNA, we call them anticodons. So this is very crucial, especially when you do translation, because the anticodons must complement with the codons, which will, yeah, so just become aware of that as well. And again, if they, for example, they give us the DNA sequence, they're telling us that it's AGGTCA, then they ask you guys, find the corresponding mRNA sequence. We know A, if it's DNA, A will bind with T, but there's no T in RNA. So A will go with U. And we know G goes with C, G is C. And now it's T in my DNA. We know the T always goes with A because there's A in RNA. The difference will be only if you are having A in DNA. So it can't go with T in RNA because there's no T in RNA. So if you're supposed to put T, for example, always change T to U. And then again, there's A, and we know in RNA, there's no T. So it becomes U. Same thing here, A, U. So become aware of such. So, okay, I've got more examples. I think you can just pause more to try test yourself. Just pause the sequence and then write the corresponding mRNA sequence. So you can pause the video and then test yourself to see if you can get the corresponding mRNA sequence. So, okay, just go straight to it. We know it's an A here. We know there's no T in RNA. So we know the corresponding will be U. Because there's no T in RNA. Again, corresponding will be U. Here, this two G we know to be C, C, G, then it's T. T we know there's A in RNA. Also there's A in RNA. So that will be what you can expect to form, which is U, U, and then C, C, and then A, A. And then this other one again. Now remember, be careful. They're giving us the mRNA now. And they want us to find the DNA. So again, it's A. We've got A in mRNA. We know there's T in DNA. So it becomes T because there's T in DNA. They're giving us U, and we know this A in DNA. They're giving us G, we know it becomes C. They're giving us C, becomes G. They're giving us U again. Remember, U goes with A. Again, they're giving us G, which will go with C. So become aware of that. Remember, it's A in mRNA, but there's T in DNA. So it becomes T here. This U becomes A. And then G becomes C, and so on and so on. So now, stage number 2, that's when the mRNA which we have formed from the nucleus will then go out of the nucleus through the nuclear pores, and then it will go to the cytoplasm, where it's going to end up meeting tRNA, which will be in translation, our stage number 3. So during translation, guys, also before I forget, become aware of the structure of tRNA, because they always give questions where they're going to ask you guys to label structures, for example. So the tRNA will come in that shape. So always become aware of the shapes of tRNA or ribosomes, because they always ask questions when they're gonna say name part labeled A, for example, or B. So tRNA will come with its anticodons. Comes like a 3 fingers, like a fork with 3 anticodons. So they're gonna go and complement the codons which are from mRNA. So the codons we made during transcription, now they'll be laying there, and then the tRNA will come with its anticodon. And guys, always become aware, they must match. If you're having a messenger RNA, for example, let's say this is my messenger RNA, it's AGU, it must complement with my tRNA. If I'm coming with tRNA of UAC, let's say I'm coming with tRNA of UAC, it must complement with my mRNA. For example, we know U, and also become also careful again, they're both RNAs, right? So must never be a T around here because they're all RNAs. So if U is coming from tRNA, it must complement with A in mRNA. Same thing, C goes with G, A goes with U, and vice versa. U goes with A goes with U. This is another example. tRNA is C, so it must go with G in mRNA. tRNA is A, must go with U. It's C, must go with G. These are the different examples that they're going to ask you. And also, they can also ask you guys to go back to DNA. Remember, here it's only mRNA. They can ask you guys to go back to DNA. They can also ask you guys to write the sequence of what you expect. Oh, sorry for my writing. My writing is not the best, guys. I apologize. So this is like DNA. It's a mess. So yeah. My N is like the worst N you guys are gonna see. Yeah. So they can also ask for the DNA sequence. Remember, that's my tRNA. It's UCA. Then we go to mRNA, which will be AGU. Now we're going to DNA. We know the A of messenger RNA will go with T in DNA. Now it's G. We know G will go with C. Now it's U here. You know U will go with A. So the DNA sequence will be TCA. So become aware. They can give you the tRNA, then they want you to go back to DNA. So you don't jump mRNA. So always become aware. If you're given tRNA, you must first go to mRNA. Then you go to DNA. You don't jump mRNA straight to DNA. No, you do them step by step. Sometimes they'll give you as a 2-mark answer. So always, if you're given tRNA, go to mRNA first, then go to DNA. But if they give you mRNA, just go straight to DNA backwards. So remember also, the tRNA that you're gonna form, it will pick the amino acids in the first step, and then only pick up one type. So the triplet base, for example, can be AGA. Then the tRNA will come and pick up those triple base pairing. It picks up only one at a time. It can't pick up more than 5. It picks up one at a time. And remember, for us to form proteins, we must at least have 2 or more amino acids, because proteins are long. They are form what we call polypeptides. So at least 2 or more proteins would be required. So, and then also remember, formation of proteins will occur in the organelle called ribosome. Because they always ask that question: which organelle will the formation of proteins occur in? Remember, it's stage number 3 of translation in ribosomes. But stage number 1, remember, it occurs in the nucleus. So transcription occurs in the nucleus. Then translation will occur in the ribosome. And then remember, there will also be cytoplasm. In case they were to point somewhere outside, there will be cytoplasm. And also remember how tRNA looks like. And also how ribosome looks like, because they can ask you to label those different diagrams as they are. So next up, I think once I've finished all these topics of meiosis and also chromosomes, your karyotypes, and your crossing over, I want to do a past paper where I'm going to focus on all the sections, which will include genetics. Then I'll just do that past paper so that I can show you guys how I normally approach those questions. So yeah, thank you very much for your time, guys.